Hi, everyone. First and foremost, thank you so much to the CNLM and just for this opportunity to get to share some work with you all tonight. You know, I think many of us here have probably had our lives affected in some way by a neurodegenerative disease. As my mom's caregiver, watching this disease unfold really gave my curiosity somewhere to go. It gave it this kind of shape and purpose. And I was left with far more questions than we had answers. And so I really wanted to contribute to this field's effort to understand the mechanisms of neurodegeneration. So zooming in on Alzheimer's disease, under the microscope we see these plaques and tangles, right? These pathological clumps of protein in the brain that we call the hallmarks of disease. But these plaques and tangles, they don't just occur in complete isolation. They exist within a milieu of cellular changes, ranging from protective to destructive. And so to really understand what's happening in disease, we've been able to look at individual cells and ask what genes they're expressing as a sort of window into their activity or dysfunction. But this gene-level approach actually misses an entire layer of biology because two cells can express the same gene but different versions of it, which are called isoforms. And these isoforms are incredibly important for cell function. Two isoforms of the same gene can even have opposing effects. One may be protective for a cell, or one might be harmful. And so I'm really interested then in understanding how the specific isoforms that cells use may become dysregulated in Alzheimer's disease. To study these isoforms in single cells, though, the standard tools just fall short. They can only see these small little snippets of the genetic message within cells, which is enough to tell you the gene, just not the specific isoform. So in my research, I use a tool called long-read sequencing, which can read longer stretches of this message to reveal which isoforms cells use in health and disease and pinpoint the ones that may be shaping disease progression. So together with all of the amazing work happening here at the CNLM and just across the field, we hope to add a few more pieces to this puzzle of how Alzheimer's unfolds and how we can treat and ultimately prevent this devastating disease. So with that, I'd like to thank my mentor, Vivek, and the whole Swarup lab, and our wonderful director, Mike, for this recognition, and just his unwavering support in every single aspect of my graduate career. So thank you.